now.
We can start the last session of the symposium. Thank you, Delvis. Thank you,
Vamos súper tarde.
for the invitation to be here today. I will co-chair this session with Dr. Santiago Ponce. Thank you to the technical staff for allowing us to be here and all the attendees to stay still for this session. We start about the biology of bee specifics with Dr. Jair Bar.
Okay. So my name is Jair Bar. I'm from Sheba Medical Center in Israel. And I want to thank Delvis and the scientific committee for inviting me to participate in this fantastic conference. And
Yeah, you have that.
Can you hear me? No? I was asked to discuss the biology, basically, of the bispecific antibodies. And these are my disclosures. And bispecific antibodies are not necessarily bispecific, they're potentially multispecific. There's a trispecific and quadrispecific antibodies. So, but most of them are bispecific. And when we think about bispecific antibodies, I think the most prominent and interesting example is the T-cell engagers. But there's also NK cell engagers which are in development. And in principle, you can envision any immune cell engager as something that can be produced with the current tools that we have, which are really sort of science fiction when you think about it. And there's a lot of interesting data that suggests that other immune cell types should be targeted to the tumor. There is this interesting publication about intratumoral immune triads of CD8, CD4, and antigen-presenting cells that have to be in a complex for the immune activation to happen. There is this data from Ido Amit's lab at the Weizmann Institute that did a very interesting analysis, single-cell analysis of cells that are interacting and demonstrated that CD4 helper T cells interacting with dendritic cells are critical for the immune response. This is data from our lab where we analyzed with a 40-plex proteomics analysis the cell types that are seen and their locations, and we found plasma cells and B cells to be most significant for the response to checkpoint inhibitors, and their location has to be within basically the TLS. So potentially you can envision also B cell engagers or plasma cell engagers, and many options are available, and there's a lot to study here. So these types of cell would be the trans-targeting, so targeting two different cells and bringing them together. I'll actually be talking mostly about cis-targeting, so targeting two epitopes on the same cell or targeting an epitope on the cell and a soluble antigen near the cell in the microenvironment. And basically, we'll start with the history. The first B-specific antibody to be approved is actually a drug for hemophilia A, so substituting for the missing factor 8 and bringing together two proteins on the cell membrane and activating factor 10 thanks to this. Going back to oncology, the most interesting example, I think, is the PD-1 bispecific with TIGIT or CTLA-4. And I'll be talking about these, specifically PD-1 CTLA-4, because they demonstrate several very interesting mechanisms of action that relate to bispecific antibodies. So first of all, if you think about targeting PD-1 and CTLA-4 with a bispecific antibody, you have to see that these cells exist. So that was the first stage of the research done in the development of these dual tools, and they do exist. And next, you have to ask, is CTLA-4, where is anti-CTLA-4 active if you want to add it to anti-PD-1? So this is work from my lab where we used ex vivo treatment of cancer tissues, and we demonstrate that anti-CTLA-4 has activity within the tumor tissue. So this is tumor tissue. This is not peripheral lymphoid tissue like what was thought initially that anti-CTLA-4 is active at. Initially, the idea was anti-CTLA-4 activates the antigen-presenting mechanisms in the lymph nodes, but this is activity within the tumor tissue only. And the other thing when you think about B-specific antibody is whether you are activating the same mechanism in the same cell, and is that important? So we know that anti-CTLA-4 works through CD28. So CTLA-4 inhibits CD28. Anti-CTLA-4 allows its activation. And in the last few years, it was discovered that PD-1 also inhibits CD28, and inhibiting PD-1, PD-L1 interaction allows CD28 to be activated and to activate the T cells. So both of these mechanisms of action, anti-PD-1 and anti-CTLA-4, converge to the same signaling mechanism within the T cell. So that means it makes sense to target both of these mechanisms on the same cell. The other issue is if you're trying to target the anti-CTLA-4 activity to the tumor tissue, what does it mean in terms of activity in the peripheral tissues? So actually, it may reduce the immune-related toxicities. That's been shown in a mouse model, and there are now drugs in development that allow CTLA-4 activity to be limited to the tumor microenvironment based on the same idea. And this is a B-specific PD-1 CTLA-4 volrustomig, where you can see that this B-specificity of the drug targets it to the tumor location. And the other important issue to discuss is the cooperative binding. So cooperative binding means that you have this B-specific antibody targeting the more abundant target first. It would bind it first because it's more abundant. And then the less abundant target would be bound to a higher degree of affinity because its concentration is higher within this microenvironment that was created relative to the affinity if this was only in the diffusing in the extracellular space. And you can see this was demonstrated in this study. You see the blue curve. This is the saturation of the CTLA-4 receptor. And you see when you have both of the targets on the cell, when volrustomig is used, there is saturation of the CTLA-4 at a lower concentration. This is relevant in this case only for the CTLA-4, not for the PD-1, which is has the higher concentration. And this was demonstrated also looking at comparing the volrustomig to each of these antibodies separately. So again, you see that the blue curve showing saturation of the CTLA-4 at a lower concentration when you have the B-specific antibody versus having two different antibodies. And this was shown also for other B-specific antibodies, the amivantamab in this case. So this is cooperative binding. The other issue is internalization. So CTLA-4 undergoes normally rapid internalization. You can see in this picture that CTLA-4 has a fluorescent tag, and this fluorescent tag disappears after a few hours because it undergoes internalization. On the other hand, PD-1 does not undergo internalization unless you treat the cells with a B-specific antibody. And you see that in this case, PD-1 undergoes rapid internalization together with the CTLA-4. And this study went into more details, and the internalization of the B-specific antibody depends on the ratio of expression of the two different proteins it targets and whether the internalizing one is more abundant or not, and probably many other factors. So several additional examples of mechanisms. So that's an NRG1 targeting drug that was mentioned, which is a B-specific antibody, functions by a dock and block mechanism, so binding the HER2 and blocking the HER3 from binding the NRG and from activating the survival downstream mechanisms. This was also mentioned, ADC, so just as an example, ADCs can be B-specific and potentially have enhanced binding, enhanced internalization. So a lot of mechanisms can be at play here, which are very relevant for the ADC activity. The issue of biparatopic antibodies, so binding two epitopes on the same protein, means that they form complexes, and in some cases, multi-molecular complexes. Here it's illustrated, you have a cap or microclusters of the cells, and this is creating unique molecular structures, and in this case, they have shown that this activates complement-dependent cytotoxicity. So receptor clustering is another mechanism. Other mechanisms at play, potentially activating proteolysis of your target of choice by co-binding of the ubiquitin ligase to your target of interest, causing it to go proteasomal degradation. So this is in development. Potentially, you can cause dephosphorylation of your target of interest by enforcing phosphatase to be in proximity with your target. In this case, CD45, which is a phosphatase being brought together with PD-1, dephosphorylating it, and then inhibiting its activity, regardless of binding any receptor, any PD-L1 in this case. So receptor dephosphorylation. So I've discussed briefly several mechanisms of actions of cis-targeting B-specific antibodies. So to summarize, as I'm trying to keep up with technology, so I asked ChatGPT to create a mnemonic of these highlights. So this is the result I got. So SPARC-T, so something that will help you when you go back to the clinic and want to remember the mechanisms of actions of B-specific antibodies. So I looked at that and I said, I have my own judgment. I'm not taking everything ChatGPT said. So I removed the T. Spark. So we are left with SPARC. And the next stage was to validate this as a tool of learning about B-specific antibodies. And so I looked at ivonescimab, and so we can identify trying to understand the mechanisms of this drug. This is a tetravalent antibody, so there is cooperative binding. That's been proven, that's been shown. So VEGF in the extracellular liquid is in dimers. So this tetrameric antibody forms complexes and then binds the PD-1 in a higher affinity, and vice versa. When it binds PD-1, it also forms complexes and binds VEGF at a higher affinity and depletes VEGF from the microenvironment. So this is the C in the SPARC, cooperative binding, and there is also receptor internalization that has been shown. I'm not showing that in the sake of time. And there is also anatomic targeting due to the concentration of high levels of VEGF in the tumor microenvironment. I didn't find any data showing support for the S and the P, so it's not all of it relevant to all B-specific antibodies. So eventually I decided to put back the T because I think this really has a transformative potential, and these are the highlights, and I'll stop here and thank you for your attention.